Hello everyone. This week we're going to talk about control of microbe growth and we're going to discuss specifically some physical type of methods. We're going to talk about boiling, pasteurization. We're also going to be talking about different radiations such as UV rays, gamma rays, and we're going to discuss how these physical methods can go ahead and affect the microbial growth. Let's talk about heat as a physical control of microbes. There is dry heat and there is moist heat. Dry heat where you have high temperature and you expose the bacteria to high temperature. Moist heat, you are exposing you're exposing your microbes to high heat and moisture at the same time. So that's why it's called moist heat. Now, what's an example of dry heat that we use in the lab? The Bunsen burner. So when you go ahead and use your Bunsen burner, which is basically, the flame, and you go ahead and use it to sterilize your loops and your needles. So you stick your loop into the flame, and right there on the spot, you're basically killing the bacteria right away. And so this is a way for you to go ahead and sterilize the tools that you use in the lab. There are other examples of dry heat, okay? So there's something called the dry oven. It's kind of like an autoclave, but without the moisture. Moist heat is the autoclave. So the autoclave is an example of moist heat, and we definitely use the autoclave machine a lot in our microbiology labs. Other examples of moist heat would be boiling, so when you boil something, pasteurization, so when they pasteurize fluids, okay, like pasteurization of milk, beverages, etc. So another type of physical, method that we're going to talk about is radiation. There's ionizing radiation, example gamma rays and X-rays, and there's non-ionizing radiation such as UV rays. Now, if you compare gamma rays and UV rays, which one would be more effective and faster in killing bacteria? It would definitely be gamma rays because gamma rays are they're much more effective because they can pass through objects. So they can pass through plastics and basically like surfaces, and they can pass through fluids versus UV rays. They would bounce off. So UV rays, for example, would have a pretty hard time passing through a surface. It would have a pretty hard time passing through the plastic. So um, definitely the gamma rays are going to be more effective compared to UV rays. Now, these are some terms, sterilization, boiling, and pasteurization. So sterilization, that's where basically you are killing all the vegetative cells, so all the live cells and their endospores, or the endospores that are being produced. And so when we talk about you've sterilized something, that term, only use it if you are talking about, that tool where all the cells like on it has been killed and all the endospores also has been killed. So it's totally incorrect to say I'm going to go ahead and sterilize my skin. Just because you wiped your, let's say you wiped your arm with an alcohol swab, does that mean your skin is now sterile? Absolutely not. We humans cannot tolerate the chemicals, or we cannot tolerate the physical methods that are used for sterilization. When you guys wipe your arm, let's say with an alcohol swab, you are not sterilizing your skin, but what you are doing is just reducing the number of bacteria that's on your skin. Okay, so again, sterilization, it's when all the vegetative cells and the endospores are killed. And so what we use in the lab to go ahead and sterilize, the pipette, the glass pipettes that we use, or for us to go ahead and sterilize our agar plates, we go ahead and stick them into the autoclave machine, which is basically going to be moist heat. It's like high heat plus like, steam along with pressure, and that's all needed to go ahead and kill off everything. Now, boiling is going to kill the vegetative cells in about 10 minutes, okay? Why are we talking about the time? Because I want you guys to see how there is a difference between boiling and pasteurization. So boiling is going to kill all the vegetative cells in 10 minutes. Pasteurization is going to kill most of them in 30 minutes. So if you compare the two, which one is more effective? Definitely boiling would be more effective, compared to pasteurization. And so that's why, again, as I was saying, pasteurization is used, you know, um, to reduce the number of bacteria, okay? And so we use the pasteurization process, for example, as I was saying, to pasteurize some beverages. So if you think of milk, is milk sterile? No, it's not, because milk gets pasteurized, and so what we're trying to do with the milk is we're trying to reduce the number of the bacteria found in the milk, and so the milk is not sterile, but what we've done is reduce the number of bacteria so that we consumers, when we go ahead and drink the milk, we're not going to get sick because we've reduced the number of bacteria in it. That you can see that it's definitely going to take more time, to kill the bacteria compared to boiling. But we can't boil the milk, right? You're going to ruin it, okay? I mean, it's not going to become milk anymore. So there are certain beverages, etc., that you shouldn't be boiling them, right? You want to be pasteurizing them, such as, again, milk. Now, with boiling and pasteurization, you're not going to achieve sterilization, okay? Because you're not going to be killing the endospores, okay? And I mean, as we said with pasteurization, I mean, you're not even killing all the cells, right? There are still going to be microbes, right? There's still going to be bacteria in that milk, and so therefore, boiling is not a sterilization technique, neither, is pasteurization. Now there is something called the thermal death time, and that's basically when we go ahead and figure out how much time is needed to kill all the vegetative bacteria, okay, at a set temperature, of course. So with boiling, we set the temperature at 100 degrees Celsius, and with pasteurization, we go ahead and set it at 63 degrees Celsius. And what we're trying to figure out with the thermal death time is, okay, how much time is it going to take if I boiled a sample? How much time is it going to take if I set the temperature at 100 degrees Celsius? How much time is it going to take to kill all the vegetative cells? Versus if I set, again, like a sample of bacteria and I went ahead and I put it at 63 degrees Celsius, which is the temperature of pasteurization. Again, how much time is it going to take to kill the vegetative cells? And that's called the thermal death time. So how are we going to determine that? When you go ahead and inoculate your plates, I'm going to show you how. You go ahead and you take a look at what is the first time point like of you not seeing any growth. So if you don't see any growth at 15 minutes, then 15 minutes would be your thermal death time. If the first time you don't see growth on your plate is at 30 minutes, then 30 minutes is the thermal death time. So the first time you do not see growth on your plate, that is your thermal death time. So in the virtual lab, you're going to be comparing two bacteria. They're the same species and everything. So B. subtilis, you're going to have an old strain, or basically like an old culture, I should say, of it, and you're going to compare it to a young culture. Now, kind of think why we're like, why you are looking at an old culture versus a young culture. What is the main difference between the two? Think of, which one is going to be producing the endospores? Is it going to be the old culture or is it going to be the young culture? And therefore, do you think there's going to be a difference in the thermal death time between an old culture that's producing endospores and a young culture that's not? Definitely you should be seeing a difference, right? The older one, you should be seeing that it's going to take more time, right, to kill, because now it has these endospores. So we'll go ahead and take a look at that together. And you're going to expose the old culture and the young culture to both pasteurization temperatures and to boiling temperatures. And you're going to compare, again, which one is more effective and faster. Is it the boiling, or is it the pasteurization? So what you're gonna do, you're gonna be given two tubes, okay? And what you're gonna do is you're going to go ahead and you're going to, of course, like these tubes are going to have the broth, okay? And of course, you're going to inoculate it with the sample, okay? So they both have the broth along with the sample. This one has the sample of the B. subtilis old, okay, so the old culture, and then this one has a much younger, sample of the B. subtilis. And for this part right here, for this experiment, you're going to be boiling your two test tubes. So you're going to set the temperature at 100 degrees Celsius, and you're going to go ahead and place your test tubes into the boiling water, right? So you're going to go ahead, place both of your samples into the boiling water. But actually, before you place them in the boiling water, you would want to take a sample as a control. So I'm going to show you how we're going to do that. So first of all, you're going to have two plates. One plate is going to be for the old sample, right? And so this right here, you can see that it's labeled bacteria 1. So this is your old sample. And then this one is where you're going to, the second plate is labeled bacteria 2. So this is where you're going to inoculate your second sample, which is the B. subtilis young. Now, what you see right here, do you see where it says 0 minutes? So you're going to, what you're going to do is on each plate, of course, after you've labeled it and everything, you're going to go ahead and separate it into 4 quadrants. And you're going to go ahead and write 0 minutes, 1, 10, and 30, okay? Now, what those times represent is how long you're going to keep those tubes in the boiling water. Okay, so you're gonna keep the test tubes in the boiling water for about a minute. You're gonna keep the test tubes in the boiling water for about 10 minutes and 30 minutes, right? And every single time, you're gonna go ahead and take a sample and take a look at the end to see, the growth. So what is 0 minutes though? 0 minutes meaning you're not even putting your sample in the boiling water. You want to take a sample. So let me grab, my pen right here. So before you place your tubes into the boiling water, what you want to do is you want to go ahead and, well, this is bacteria 2, so you're going to go right here. And you're going to go ahead and inoculate a sample, okay? And then from bacteria 1, you're going to go ahead and inoculate a sample right here. So basically, the 0 minutes is the first inoculation before even placing your samples into the boiling water. Okay, so this is what it means by 0 minutes. You are inoculating the plate before even putting your samples into the boiling water. Okay. Now, what does 1 minute mean? It means that you want to keep your samples into the boiling water, both of them, okay? So you want to go ahead, place both of them into the boiling water, and you want to keep them in the boiling water for at least 1 minute, okay? And then after that 1 minute, you're going to take it out and you're going to go ahead and place it, inoculate it onto the plates. But if you think about it, okay, for you to go ahead and sample it at 1 minute, when you place your test tubes into the boiling water, you need to wait for at least 30 seconds, okay, before setting your timer for 1 minute. Why do I want to place my test tubes into the boiling water, wait 30 seconds before I start counting down before I go ahead and set my timer to 1 minute, because you want to give your samples a chance to even get heated, right? It's not like I'm going to put my samples into the boiling water. There we go. They are already at a boiling temperature. Absolutely not. It's going to take them, some time. And so therefore, go ahead, take your samples, put them into boiling water, wait for 30 seconds just to give them a chance to go ahead and start getting heated, and then set your timer for 1 minute. And then after, after you set your timer for 1 minute, and let's say time is up, you're gonna pull them out, okay? You're gonna pull your test tubes out of the boiling water, and then you're gonna go ahead and you're going to go ahead and inoculate your plate at the quadrant of 1 minute. And then quickly put them back, okay? So quickly then put them back into the boiling water. So if you think about it, I mean, we kind of took the sample of the 1 minute, really not at 1 minute per se. I mean, it was the test tubes were in the boiling water for about a minute and a half, which is fine. Again, we had to kind of do that so to give a chance for your sample to even get hot in the first place. Now, after that 1 minute, quickly put your samples back into the boiling water, and you're gonna wait for another 9 minutes. Now, why 9 minutes? Because these times right here, you guys, like 10 minutes, if we're talking about 10, 30, etc. Now, this is like how much total time you should have placed your sample in the boiling water. So if you already placed your sample in the boiling water for about like a minute already, then go ahead, put them back into the boiling water, wait another 9 minutes, and then that would be a total of 10, and then go ahead, stick them, take them out, inoculate, and then quickly put them back in. You don't want to take so much time, okay, guys, when you are inoculating your plates. You want to quickly put them back, okay, into the boiling water. And then you're going to go ahead and inoculate your last plate, like your last quadrant. So your last quadrant is at 30 minutes. So, so far, right, we've already put our sample for about 10 minutes already into the boiling water, right? So then you want to leave it for another 20 minutes, right? So that the total would be 30, okay? So you already boiled it for 10 minutes already. So stick them back in, wait another 20 minutes, right? That would be your 30 minutes, and then stick them, put them out, pull them out, and then go ahead and inoculate your last quadrant, okay? And so this is how, this lab is usually done. After, of course, you've inoculated all of your quadrants, go ahead and incubate your plates, and then take a look at your results later. Now you're going to do the same experiment, but this time what you're going to do is you're going to go ahead and test the pasteurization temperature. So the pasteurization temperature, if you recall, you need to set it at 63 degrees Celsius. So we usually, for this one, we use a hot plate, okay, or not hot plate, I'm sorry, like a hot water bath, okay? So we use a hot water bath, and you can see right here, we can go ahead and set the temperature, okay, to the temperature that we want. So you get a hot, hot water bath, which kind of looks something like this, and you fill it up with water, and then you set the temperature to whatever temperature you want. So right here, we're going to set it to 63 degrees Celsius. And then after it reaches 63 degrees Celsius, go ahead, take your sample, and of course your sample already has the broth along with the bacteria. And you're going to go ahead and place both samples into the hot water bath. But one more time, before you place it in the hot water bath, do not forget to inoculate your control at 0 minutes. So, and the reason why you want to have a control, to compare your results, and to compare your results, especially, when if you want to see like how effective is pasteurization, I mean, you want to see first of all that your bacteria is even growing in the first place, right? So the 0 minute is going to act as your control. And as I said, the 0 minute, you haven't even placed your samples in the hot water bath yet. Um, After you've inoculated your 0 minute quadrant, place your test tubes, both of them, at the same time into the hot water bath. And then one more time, wait for 30 seconds though, right? You want to still wait for 30 seconds just to ensure that, again, you're giving enough time for your bacteria to kind of reach, that temperature and to be accustomed to that hot, temperature that you've just placed it in. And then after those 30 seconds, start your timer for a minute. And then once the time is up, go ahead, pull them out and inoculate your quadrants, okay? After you've inoculated your quadrants, same thing, stick them back into the hot water bath for another like 9 minutes or so, and then inoculate your 10-minute quadrant, and then stick them back in after about 20 minutes. Do the same thing, inoculate your 30-minute quadrant. Incubate your plates, and then after incubation, you're going to start looking at your results. Now, we're also going to talk about UV radiation. So as we said, UV radiation has a hard time passing through, hard surfaces, plastics, fluids, etc. But that doesn't mean that the UV radiation is not effective at all. It is, and it can kill the bacteria. But I mean, if you compare it to gamma rays, the gamma rays are definitely much more effective. And how does UV radiation work? How does it go ahead and kill the bacteria? Well, the UV radiation can pass through the bacteria cells, and it will start actually damaging the DNA. And the way it damages the DNA, if you have two thymines, right, two thymine bases, what it does is it goes ahead and binds them together, and we end up calling this thymine dimer. So right here, so basically it creates those thymine dimers. You might think like, okay, what's the big deal of two thymine bases being attached to each other? Well, that's going to basically affect the DNA replication. So it's going to prohibit that part of the DNA to get replicated. I mean, if you think about it, bacteria, they go ahead and they multiply, right? And this is how they would want to, like, colonize, right? And take over, right? The host. They want to colonize the host. And so if they cannot replicate their DNA, how are they going to replicate themselves? And so therefore the bacteria dies. Now for the lab, okay guys, what you're going to do is you're going to go ahead and you're going to grab a plate, and you're going to go ahead and inoculate your plate with the bacteria sample. Okay, so you're going to go ahead, like, inoculate your plate with the bacteria sample. Of course, right here you can use the cotton swabs, okay, sterile cotton swabs, stick it into the broth that has the bacteria sample, inoculate your plate, and then what you're going to do is you're going to take that plate and you're going to actually take off the lid. Okay, you got to take off the lid, and the reason why you got to take off the lid, because you're going to expose this plate to the UV lamp. Okay, so this is the UV lamp right here, and the reason why you want to take off the lid, because we just said that UV lamp has a hard time passing through surfaces such as plastic. And so if the lid is on, the UV light is just going to bounce off, okay? And so therefore, make sure that you do take off the lid. So you want the UV lamp to directly, you want basically the agar, right? That now you have the bacteria inoculated on it to be directly exposed to that UV light. Then what you're going to do, you're going to take a card, okay, and you're going to go ahead and you're going to basically like hide half of the plate. You're going to put it on half of the plate like so. And so why are we doing that? Because one more time, this is going to be kind of like a control, when the UV light hits the, like, that paper, what's going to happen? It's going to bounce off. Okay, and so you want to see that difference. You want to see the difference between the sides, okay, like this side of the plate, what's going to happen to the bacteria growth compared to this side of the plate that was exposed directly to the UV light. Okay. Now another thing is, you're going to go ahead and play around with the times. You're going to, set the timer for like 5 minutes and see what happens when you expose the plate under the UV light for 5 minutes. Then try it out, so you can play around with the times and actually try it several times with different plates. So, set one plate to be exposed for the UV light for 5 minutes, set another plate to be exposed for 1 minute, set another one to be exposed, under the UV for 30 seconds, okay? And right here, see notes, that's like the following slide where I'm gonna talk about, okay, will you see a difference? Will you see a difference in terms of the bacteria growth being like, or the bacteria inhibition, if you place it under the UV light for 5 minutes versus a minute versus, like, 30 seconds, etc. Another thing actually to talk about is also the distance, okay? So it does make a difference, okay, guys? Maybe I should go with a different color right here so that you guys can see. So right here, okay, the distance of how close am I placing my UV lamp to my agar plate does make a difference. The closer you expose your sample closer to the UV light, the better, the better results you're going to see in terms of the bacteria being inhibited. So you can go ahead, not with this virtual lab, but I mean, in reality, you can go ahead and play around with how much distance, okay, you want to place that UV lamp onto your sample. You can get it closer, you can, put it further away, and go ahead and check out what's going to happen. So let's take a look at, the results that we expect to see. Now we expect to notice that the length of the UV exposure. So basically, how long am I exposing my bacteria to the UV light? The longer you expose it to the UV light, the more killing. Okay, so the more bacteria are going to be killed, and that is a directly proportional relationship. The more I expose it to UV, right? So the more I expose it to the UV, the more killing is going to happen. So that is definitely a direct proportional relationship. Another thing is the distance, right? Are you putting your plate very close to the UV light, or are you putting it further away from the UV light? So this one is an indirect proportional relationship. Why? Because the more distance, okay, so meaning my UV light is not that close to my bacteria sample, the less bacteria is going to die. And so that is an indirect proportional relationship, right? The more distance, the less killing. That's an indirect proportional. Another factor is, okay, after you've exposed it to the UV light, did you expose your plate to visible light or not? And we're going to explain why visible light is important, but it turns out that when you expose your if you expose your bacteria sample to visible light, which is basically regular light, right? Lamp, basically like a regular light. It turns out that if you expose it to visible light, after exposing it to the UV light, less bacteria is going to be killed. And so that's another indirect proportional relationship. Now, it turns out that bacteria, they actually have two repair mechanisms that can help them repair DNA damage caused by the UV radiation, because we just said that UV radiation is going to penetrate the cells and it's going to cause the thymine dimers. So bacteria have two ways to repair that damage. Um, Bacteria have an enzyme called endonuclease. This enzyme, it goes ahead and repairs that damage that the UV radiation has caused. It is effective, but it's pretty slow, okay? And it can work 24/7 though. Um, if the bacteria is in complete darkness, or if it's exposed to like natural light, the endonuclease is going to go ahead and work. But again, it's a pretty slow, not that efficient of a process, okay? Now, bacteria also have another enzyme called DNA photolyase, and this one is fast and efficient, process in repairing the DNA damage, but it's only active under visible light. So it's only going to work if the bacteria is exposed to light, meaning that if the bacteria is in complete darkness, then the DNA photolyase is not going to work. So if you think about it, if you have, let's say, your bacteria on your agar plate and you have it incubated in the incubator. Now there is no light in the incubator. So the bacteria in the incubator, it's basically in complete darkness. So if the bacteria is in the incubator and it's trying to go ahead and repair damage caused by the UV radiation that it was exposed to, then it's only going to use the endonuclease. But let's say you have your agar plate that has the cultured bacteria, and let's say you've exposed that bacteria to UV radiation, and then you went ahead and you exposed it to natural light, then what's actually going to happen is both endonuclease and DNA photolyase is actually going to work, because both of them can work, you know, during daylight, or basically when the bacteria, I mean, it doesn't have to be daylight. You can expose your bacteria to a lamp and that would be light. So both of them though will go ahead and work if the bacteria is exposed to light. So you can see that the bacteria that are exposed to light, they're going to be able to repair the damage faster compared to the bacteria that are in complete darkness. Now, it turns out humans, we can also repair DNA damage that we also might, experience. How do we do it? Well, we have the endonuclease enzyme. Okay, so humans, we have the endonuclease. We do not have DNA photolyase. Okay, so we humans do not have this enzyme right here. We only have the endonuclease. Um, and then you guys are going to have like a couple of videos that are pretty helpful, so go ahead and watch them. And then of course you will have the virtual lab to complete. You will go ahead and take a look at those results. So right here you have the B. subtilis old. So you have an old culture of the B. subtilis, and then you have here a young culture. Now you have this old culture. You've boiled it, and then another old culture, you went ahead and you've put it in the temperature of pasteurization. So you're going to go ahead and take a look at the growth results that you get on the plates and try to figure out the thermal death time. And then for the young culture, same thing. You have a young culture that was exposed to the boiling temperatures, to the 100 degrees Celsius, and you have a young culture that was exposed to the pasteurization temperature, which is the 63 Celsius. One more time, take a look at the growth, okay, on your plates and try to figure out the thermal death time. And then also try to figure out, okay, you know, is there a huge difference that you see between the old culture and the young culture? So compare the results, okay, for the old and the young, for both when they were exposed to the boiling temperatures and the pasteurization temperatures. And then right here, you know, for the UV radiation experiment, let's say you have a culture of E. coli and you went ahead and you, let's say, exposed the E. coli to UV radiation for 0 minutes, meaning they were not exposed at all, and then you exposed them for 1 minute, 5 minutes, 10 minutes, and then again, you're going to go ahead and take a look at your results. These bacteria right here, they were, you know, exposed to the UV radiation at a far distance. E. coli right here was exposed to the UV radiation at a close distance. So compare the results and see if the, you know, the distance of like, you know, the UV radiation exposure makes a difference, okay, in killing the bacteria.